Okay, hello everybody. Welcome to part 3 of RNA-Seq with DNA Subway. I'm Jason Williams coming to you from the DNA Learning Center at Cold Spring Harbor Laboratory. This is the last of the 3-part series, so wherever you're watching it, if this is the first time you're seeing me for this, then you want to go back and find the other videos on the DNA Learning Center YouTube channel. Let's start, as we always do, with a few slides just to introduce and to recap, and then we'll go into everything else that we have planned for today. Okay, wonderful. So this is part of the DNA Learning Center's DNLC Live. So we really love and enjoy your feedback. You can leave us feedback in the YouTube video comments or find us through social media. I'll mention that in a moment. But do make sure you come to dnlc.cshl.edu and look in our live section because you'll also find associated with this video resources, including a worksheet that you may want to follow along. It's got some questions that can pose you to think. And like I said, these are our social media handles, so feel free to go ahead and take a look at those, leave us feedback, tell us what you'd like to see, or give us some comments. This is an actual kind of course. It's intended for undergraduate, so on a 200 level and up, you probably will have needed that first year of biology and maybe even an elective. If you're taking advanced biology in high school, AP, you've completed that, you probably should get something out of this course, but there is a little bit of an increased difficulty level. This is the last of our 3 sessions, which have been actually more like an hour, but in any case, I'm sure that people can watch it at their leisure at a pace that works for them. One of the great things is that there are exercises, or we're doing exercises, so you can actually follow along on the online bioinformatics tool, DNA Subway. And like I said, the slides and the sheets are available. Now, we've had several learning goals to understand some of the background of what an RNA-Seq experiment is and what it can actually tell us. And then we wanted to really learn how to use several tools. And we're going to go in and try to understand a little bit about them. And we have done that. Now we're coming up to sort of the high point here when we're going to actually look and see our results for our differential analysis. As I mentioned, we're using DNA Subway, which you can find at cyverse.org or Google DNA Subway. But in order to actually do the exercises, you need an account. It is free, so go ahead and sign up for an account, but then you can follow along. Okay, so we're finally up to the highlight, the climax here, differential abundance or expression. We need to parse that word or those terms a little bit, but that's where we are at this point. So let's review what we've done to get here. Let's go in a little bit deeper as to what does it mean to have differential abundance or expression. And then we're going to visualize and explore the results and tell you what we get from that. So to review RNA-Seq, these are the key bullet points that I've referred to every lecture. RNA-Seq allows us to measure the transcriptome. So we're looking for all of the mRNA, usually messenger RNA transcripts. And we use the abundance of those transcripts as a proxy for some cellular process. So in other words, if an RNA transcript is more abundant, then there's an inference, not always a true one, but there's an inference that perhaps then that gene is active. And it may not even always be a gene in the terms of at least a protein coding gene, let's say it that way. And in an RNA-Seq experiment, we're usually categorizing these differences or I guess we could say describing these differences between samples. And they could be cancerous versus non-cancerous, or it could be at different developmental time points or for different reasons. There's a lot of text up on here. I'm going pretty quickly through this, but go back to the other videos and you can see that in more detail. One of the key concepts that we need to understand, I'm going to drill it in one last time, is variation versus difference. This is what makes, one of the things that makes an RNA-Seq experiment difficult, and we're going to come back to this in a few minutes when we actually really see what we're doing. But the key thing is that if we have an observation, if we make a biological observation, like in this case, height of individuals, we have to realize that in biology, the quote is sometimes, biology is messy. And so there is not just one height for a biological female that is a height that's typical of females. There's an average. You could say, as it says on this sort of chart, that it's somewhere close to 160 centimeters, but you could have women that have a greater or lower height, and the same thing for men. And one of the things that we're trying to tease apart often with math is to try to figure out, is it more likely, given a certain height, that our observation came, in this case, from a male versus a female. So that's just a distribution. That's a concept that we need to look into. I've explained it to some extent, but look at those other explanations or find some ones that click for you. But it's something you want to understand, that it's part of doing this experiment. Once again, we're working with Zika virus, or rather human neural progenitor stem cells that have been infected with Zika virus. And I explained in the very first video some of the effects of that infection. And obviously it damages those neural stem cells and leads to other significant brain damage. Okay, so far in DNA Subway, we've done a number of things. We've talked previously about how we get that data, how we manage it, how we clean it. And then our last episode, well, here in sequence quality, we say that before the last episode, this is the first episode, we talked about the fact that even though we end up with millions of reads, and each one of those, you know, we may have many hundreds of base pairs per read, really every single nucleotide call or observation of a nucleotide has a score attached to it, a Phred score, which gives us the idea of, was it a good quality read? You need to have good data in any experiment in order to trust that the results are good. And so we talked about how we evaluate that. Another key concept, it's going to be a key concept again, is the idea of alignment. The thing I'm going to explain in the next slide is that we are mapping these individual sequence read. We get 100 base pair, 150 base pair, whatever the length is, reads of DNA sequence, and then we need to find out where in the genome those reads came from. And since we're taking those observations and they are basically corresponding to the amount of mRNA that we got in our preparation, then if we have more reads, then we're assuming that the gene, if it is a gene that we're looking at, may be more active, whatever that means. Sorry to be ambiguous, or but it is a little bit ambiguous. It doesn't always have to be a gene, and active may or may not be the right inference. So because we have more of an mRNA, is it because that our mRNA, does it actually get made into more protein, or is it going to be degraded by another protein, or is that mRNA particularly stable or not stable at all? So those are other caveats. We don't have to worry about them too deeply, but if you're a researcher, then you better well figure out what you can figure out in terms of the observations that you're making in your particular system and the genes that you care about or the loci that you care about. But this kind of sums it up nicely. We get samples, like in this case, sample 1 and 2 from our sequencing machine, and each one of those little reads, we need to map them, and that's a computational process, to a location, and then we do some kind of counting. And counting needs to also, there's a bit of math that actually helps us to understand what is a fair count, and we'll talk about that. So to do that mapping, or to do that, get that count data, we looked at this, or we used this software called Callisto. Callisto has been around for a couple of years now, but it's the reason why we used it is because it's extremely fast. And also, not only is it fast, but it is very accurate. So those are two good things. Often you can't have both, but in this case, we do very well with both of those characteristics. One of the problems that we had is that we're looking at a transcriptome, and so we need to figure out if we have a transcriptome, which is the sum of all transcripts, how do we match those sequence reads? And we talked about last time this computational, the de Bruijn graph. With the de Bruijn graph, you can go ahead and take a read, which is here in black. This is a figure from the original paper, so to really get an understanding, you might have to read that. But if you have an actual read, and then you have transcript, a pink transcript here, a blue transcript, and a green transcript, your question is, which of these transcripts is this read compatible with? And by eye, what I hope you're getting even before you look at the Bruijn graph is that, well, the green transcript up to a point kind of matches, but then the green transcript ends, right? And there's a gap here. Maybe there's a long intron. And that's not what we see with this read. So this read better explains the pink and the blue much more so than it explains the green. And so you need to do this, but you're doing it literally for every single read. So it's an interesting thing. And then reads can map to more than one transcript nearly flawlessly. And so that's another problem, whether it's genome duplication or other things, but we don't need to go that deep. This is just another way of explaining it, that we're trying to do that mapping to figure out what's the best possible match. So we did the pseudo alignment, and last time we told you that we have, for our data sets, since these are pairs of sequencing reads, a forward and a reverse, we have the mock sample, which is, these are cells that are not infected or not challenged with Zika virus. So we have sample 1, replicate 1, that's what this 1-1, and sample 2, replicate, or mock, you know, this is replicate 2 of this sample 1. The same thing, we have 2 replicates for cells that were challenged with Zika. So what we need to do now is take a look. We briefly took a look last time, but now let's really go to DNA Subway and make sure we understand what we are looking at. So to do that, I'm going to go ahead and get to share my DNA Subway screen. Okay, and I'm going to go ahead and log in. Okay, so we are in DNA Subway. I'm going to go to a project that I've now done. I don't need to take the survey. And when we do this Callisto alignment, in my case, I did it more than once. You do want to view the results. And you also want to check things. If you click on this number sign, you want to see how many reads were aligned. So we started out with 7.6 million reads in this individual sample. And we actually need to do the alignment for every single sample. And about 6 million of them actually map. So that's a 79% rate. And that's what you expect. You rarely would really expect a 100% rate, but you expect a pretty good, somewhere between 80 to 95, somewhere, you know, I'm not setting a rule. If you got a really low rate, then you have to be careful that you haven't made a mistake somewhere, like you chose the wrong transcriptome or you need some other error. As we saw last time, you click the folder and it would actually take you to the data on Cyverse, which you could download and you could use in other programs. But I'm going to go ahead and click on download. And what it's going to do is download a essentially big spreadsheet. And we're going to just take a look at that and also define some terms. So I need to choose how I open this. So it's a TSV file. It's going to open it up with Excel. And now that I've downloaded, I'm going to go ahead and share that. Okay, so now we're looking at Excel. I'm zoomed in here a little bit. And I'm going to get this a little bit. Now, if I recall, also comes out with a little bit of garbage. Is this true for this one? Hopefully, maybe not. Maybe so. Yeah, I remember that there was some HTML stuff that we don't need because I want to do some data sorting. So I'm just going to get rid of that. We don't need that. Okay. Now, what we're looking at here is some data. I want to define this for you, but I want to have this in Excel just so I can sort things. We're going to see another way to play around with the data, but we need to explain what all these columns mean. So in order to do that, I'm going to go back to our slides where I have tried to parse that for us. We'll take another look after we understand that. So there are several columns, and the first column is something called the target ID. And target ID is an identifier, so it's a unique identifier for the transcript. Genes, of course, you know, usually have names, especially if they've been worked on. But in order to, sometimes what happens is they have many names, which gets confusing. So they have a unique ID, one and only one for any particular transcript. So that came from Ensemble, which we talked about last time, and which we'll be going to again. In fact, I guess we're going to go, well, let me go through the rest of the column and then I'll go back to Ensemble. We also have the length, which is how long that transcript was, the total number of nucleotides for that exon. Then the next one is something interesting, and it's called the effective length. So it's the length of the transcript that was sampled during the mapping. And the little fine print says that in the sequencing, when we do the sequencing, this is a very subtle thing, but we rarely get full mRNA transcripts when we prepare our sequencing library, and I discussed that a little bit in the very first episode. So we actually usually chop up the DNA even before, forget about sequencing reads, when we're preparing our sequencing library, we usually chop it up to something like 300 base pair fragments. So there is some little manipulation or accounting for that in the effective length of what we actually have sampled. And then there's also, given that information, the count data is an estimation of the number of reads that have mapped to the transcript. So if it's 0, it basically means that we didn't see that transcript. We didn't see any reads mapping to that transcript. Maybe that's a gene that was not expressed. Okay, so we're going to come to normalization, but let's go back to Ensemble and to our data set with just those things explained. So here are our target IDs. You could see, for example, the length of these transcripts here is pretty small, right? So it's not surprising for these smaller transcripts. Maybe these are things like microRNAs, which can be around this size. It's not surprising that we don't maybe see very much. But we can do some filtering, and hopefully, although this is not a lesson in Excel, you might know some details. If we go to data and sort, we could sort by length. Maybe we want to sort by length, and let's go from the largest to the smallest. Okay, so here is a gene, and obviously it's quite long, and there were 42 reads that map there. Here is another gene or transcript. Now, what I'm going to do is I'm going to take this, and I'm just pick that one purely at random, I hope, and go back to web browser. Now I've already gone to the Ensemble website, and many of these links are in our packet on DNALC Live site, so go check that out. And if I paste in here one of those IDs and click go, what's going to happen is Ensemble is going to find it. And if I click on it, it tells me that this is a gene called SMAD2. And it's pretty cool because I can, find out where this gene is located, and also a little bit about the structure of this gene. So if we're on here, if you read through these details, if you click on the name, it's also going to open up a little bit about this. So SMAD gene family member, and you can go ahead and look and see a little bit about what this gene does or what's known about it. So you could just go through and really pick out, this happens to be on chromosome 18. So all of this data is here, and you can figure out a little bit more about any one of your transcripts. But for right now, one of the questions is, why should I pick that one or any one of them? How do I actually know? But certainly it's pretty cool, I think, that you can go ahead and just by that look at it. So the thing to understand, going back to looking at that spreadsheet, is we have that gene identifier. We have an idea of how long it is. Now it said, and I've already forgotten which one. Let me go back and look. This was, well, it should be in my clipboard here. 16010. So going back to Excel, so 160.10. It said that this is 34,000 or so nucleotides. And if I go back to my web browser and I'm looking at it, it doesn't give me the count there. But by eye, well, that's one of them. these are different isoforms, so there's different versions of this gene, which are probably alternatively spliced. I was just looking to see if it gave me quickly. Yeah, that smaller one was 12,000, and yeah, 34,000. So we're getting, all of these things lining up the way that we would imagine. Okay. So the next thing I want to explain is really this other thing called TPM, because this is what we're going to eventually use in order to actually look really at the differential expression. But let's go back to the slides to explain what that last column is. Now, in order to talk about that, we're going to talk about a concept called normalization and use some illustrations to help us understand that a little bit better. So this last column stands for transcripts per million. And the definition of it's what we call the normalized counts based on the length of the transcript and on the total number of sequence reads. So there is a formula that defines this. And this term in the formula called A is the total reads mapped to a gene. This is actually a typo. That should be, fix it now, superscript. Sorry about that. Total number of reads mapped to a gene divided by the gene length. There's some terms here, some subscripts, they're not really shown, but basically for every single transcript, you need to calculate this A term. There's a lot more math ahead, but let me just reduce it to a question and some things that can help us understand what we're seeing. So here is gene A and gene B. We've got exons, and then the little lines in between are introns. So my question to you is, which of these is longer in base pairs? So by eye, it's quite clear that they are of different length. And let's just say that gene A is longer, right? And in fact, I'm just making up some numbers, but they'll work for us. And gene A, I'm saying, is 3 times the length of gene B, 300 base pairs. Now the next question, once we understand, okay, genes, in our case, transcripts have different length, great. But the next question is, which of these has more reads? So if we had sequencing reads, as we've seen before, done in black, they map to, they might match completely over an exon, maybe they overhang an exon, maybe if they cross an intron, then usually that line there just means there's missing data. But obviously the reason why is because transcripts don't have introns, so that's what we're seeing here. But now my question here is, which one of these, gene A or gene B, has more reads? So I don't really need you to pause the video to count, but if you were to, you would see that there are 27 reads mapped to gene A, and there are 16 reads mapped to gene B. Okay, great. But that is not really a fair count of expression because they are different lengths. And so the key thing about normalization is that we want to take into account the fact that because genes vary in length, then we need to sort of take away some of that for genes that are really, really long, which are going to then thereby have more reads, and also give a little bit more credit to genes that are short because they are shorter. And so they won't have as many reads, but they're not as long, so they couldn't collect as many. So if you do that, and if you just divide, not that this is the only or entire way of doing this, but you get the idea that you can come up with a ratio, and then the ratio for gene A is lower than the ratio for gene B. So it's sort of punching above its weight, even though by eye, just the raw number of counts might conclude that gene B is lower in expression level. If we normalize and take into account that it's also shorter, then we can actually get a better understanding of really which has the higher level of gene expression, which in this case, we refer to that speed. Now here's another question. Which one of these genes is more highly expressed? Very similar setup. You have genes A, B, C, and D. You have a number of reads in them, which you could count. Although by eye, to me, certainly A looks like there's not as much going on as C and D. Now this flow cell is actually an Illumina flow cell. This is a glass slide, although a very sophisticated one where the sequencing takes place. But they're different. So when you do, every time you do a sequencing reaction, how things are prepared and how things are run will lead to slightly different results. So you need to ask a question, how many total reads did you get from a run of an experiment? And then if you look, in flow cell 1, imagine that our experiment yielded 10 million reads, but experiment 2, we got 20 million reads. So now the fact that gene C and D appear to have a lot more, maybe double the gene expression of gene A and B, has to take into account that, well, there were a lot more reads overall taken, forget about which gene they map to, but there's a lot more data observed on flow cell 2 than on flow cell 1. So we need to, again, normalize how many total reads are we actually looking at. So all of those factors go into normalization and then generate this corrected normalized counts called transcripts per million. And so it's a more accurate thing. Now what we're going to do in this last section of this presentation is talk about how do we actually understand those differences, quantify those differences, and in fact visualize those differences. So there's another tool that was made to go with Callisto, and that tool is called Sleuth, and that's what we're going to use. And remember, again, we're looking to find the true differences between samples while subtracting differences that don't matter, like variation, because you know, you perform the experiment on a Tuesday and I did it on Wednesday, or one flow cell got a little bit more reagent and so it generated more sequencing reads. How do we subtract that? And then the hardest one is that, you know, biology is different, and so, you know, one mouse had a cold one day, but, you know, that has nothing to do that we were looking at, you know, cancers. So this game of spot the difference that we alluded to in the original slide now looks something more like that. We need to take each of our 4 data sets in that case and find the differences in those data sets, accounting for what we discussed so far and even some more things. Now, again, you'll need to go into the paper. There's a lot more math than I'm sparing you, but sort of what I said, there's biological variation, technical variation, all of those types of variation that go in to make what we use, which is a linear model. And then you'll need to understand a little bit of statistics to think about what it means to do a linear model and to subtract things out. Since I've spared you the math, I won't spare you this meme, which I found today as I was preparing the lecture and I thought was cute. The message to you is, if you're a biology student and you have the chance to take an extra linear algebra course or a stats course, then think about doing it, because it's one of the things that will certainly help you to get your head around problems like this. Even though many of us came from biology, we also want to be able to do some of the math. Okay, so what we're going to do now is take a look at these results as they appear in DNA Subway. So I'm going to go back and bring up DNA Subway. I'm going to close this window and go to Sleuth. And this is the 380.025 job. I'm going to go ahead and launch an app. So this analysis is using a computer language called R, which is a popular language for doing statistical programming. And it's in the form of an interactive app. So you don't actually need to use R in order to make use of the application. It's really nice. Although if you downloaded these data and then wanted to, you could use R to go ahead and study it further. It's a rather large data set, so it's just going to take a minute or so for this all to load. So we'll just wait a moment for that to happen. Okay, so it's loaded, and what we see here is a column, and some of these things are similar, but some of them are different. So we do have the target ID, but we also have this thing called a Q value, a B value, and also this EXT gene. So what are those? Well, thankfully, they are explained on the left-hand column. So just like before, the target ID is our ID, and you can also add or subtract these columns however you would like. And really, to understand what some of these columns mean, like the variants, technical variants, all those different things, go, you'll have to read the paper. But what we have in front of us is sufficient. The Q value is a statistical test. It's a corrected statistical test, which is trying to tell you the significance of the observation. And what is the observation? Actually, before I explain the Q value, I should explain the B value. So the B value, the beta value, is an idea of trying to explain to you what is the fold change difference between the samples. In our case, if it's asking the question about this, let's say this first gene that happens to be there, HYOU1, what is the fold change between a Zika infected cell and a control mock cell? So in this case, it's saying that there's an increase of gene expression. That's a 0.9 fold increase. So a little bit more gene expression. Whereas in some cases, this gene DHFR is minus 1.7. So it's 1.7 times less gene expression. And when you're looking at these fold changes, then this Q value is trying to give you, or trying to say, what is the chance that you would make such an observation just by random chance? So what's the chance that this observation is just a fluctuation? And so the smaller this number is, the more confidence that you have that this observation is probably a real observation. It would be akin to, if we go back to our slides, although I didn't plan it, but this is already written, isn't it? If we go back to the slides and go back to the slide we had earlier, it's sort of actually like this. So what is the chance that you find a female who has a height of 195 centimeters? Well, the area under the female curve doesn't even reach to 195. So that means it's highly, highly unlikely that you would make such an observation and that that height doesn't belong to a male. So that's what it's kind of telling you. If I say, what is the chance that you'd observe a female that has a height of 175, well, it's still rare, but it's out here on this end of the bell curve, and maybe that's something that only happens 3% of the time, okay? So this is what this value is telling you. The smaller the value, the less likely it is that that value has occurred by random chance. So what can we do with this data? Well, there are a couple of things. I'm going to take you through a little bit of it, and we're going to explore with the last few minutes that we have left what the original paper found and see if we found the same things that the original papers have found. There is a graph here called a bootstrap where you're able to look at the gene expression level or the estimated counts for our samples. They're colored, the mock and the Zika. If you look, we have two replicates, and the replicates are not necessarily the same level. You probably wouldn't expect that. But their error bars that are here, these quartile ranges, show that these are basically the same observation. So that's good. You want them to agree. And what you can do is you can choose, and we'll come back and intelligently choose something. You could choose different transcripts at random, and it's going to go ahead and give you a graph, and you could see what that graph tells you. You could even go ahead and save that image if it was a gene that you know you're interested in for some reason. There are some other things which I'm not going to go so much into for the sake of time today, but there's a principal components analysis, and what you're really looking to see there is that there's a correlation between, or that there is a grouping between the mock and the Zika samples. One thing was not an outlier. There's also this volcano plot, which allows you to look at individual transcripts and see whether they go into increased expression off to the right or decreased fold change off to the left. And you get an idea of some genes are not significantly called, so those are in black and some are red. This other principal component tells you which genes are mostly influencing that PCA analysis, which again, I don't have time to go into this second. And there's also a heat map that gives you a little bit more. So let me go back to the differential analysis. And what we want to do here is, well, let's just click on the results. And then what I'm going to do is, I think I want this, I want the fold change. Is there anything else that I want? I think that's all that I want. What I'm going to go ahead and do is click on download so that I can sort this a little bit in Excel. Okay. And I am going to go also and share my Excel. Sleuth results, I think. Okay, so this is basically the same thing. It's just in Excel, and there's some things that I want to show you how to do here. Because I want to sort by more than one thing. If you go back in the DNA subway, you can sort by multiple things. What I'm going to do here is going to select my data set, and I want to look for the genes that have the most change in gene expression. So I am going to go to data and sort, and then I'm going to sort by fold change, which is B, and then from largest to smallest. Okay, so there's a 6-fold fold change, positive. And the next thing I want to do is, if I could look at this gene, okay, happens to be a p-value that's low, but maybe not all of these are low. So what I'm going to do is get rid of genes that are not significant. And usually the cutoff is 0.05. So what I'm going to do is filter. And advance. Actually, what I need to do is maybe sort. Let's see, there's a lot of genes we're looking at. And I usually do this in R, but I think I will be able to figure out. What I did the last time. What I'm going to do is just, okay, that's what I wanted to do. I need things that are less than or equal to 0.05. All right. So what I've done here is everything that's in this gene list, I don't know, I didn't care to look, but everything that's in here or that I've sorted by is giving me these more significant genes. And I don't know that I've really gotten rid of all of them, but I've at least filtered them in a way that I have the top ones on top. So let me just make sure I'm doing that correctly. Oh, 0, so 0.5. I was like saying, we didn't get rid of all of them. That looks better. Always good when you do a sort to actually make sure things turn out right. Yeah, now I'm getting to the end and everything that I see looks like we really have genes that are 0.5 or less. So theoretically significant genes. Now what I want to do is take a look and see what some of these genes are. And I could do that one by one. So let me just take one of them, the first one, CELF2, and I don't happen to know what that is. But as you know, we can go ahead and take a look on Ensemble. We can go back and I can search in human, I can paste it there. And see what it tells me. Okay. And if I look at this, it's going to tell me a little bit of information about the gene structure. And then I may want to go and find some additional information about this. And you could even go ahead very simply. Well, if I go back to gene versus transcripts, it gives you a tab here on the left-hand side. It gives you some of the things that it's with. And then if I went ahead and just Googled it, so it's involved in alternative splicing, and you go through and you can see some additional information about what it is. But what I thought would be interesting is to look at the original paper. So let me go ahead and do this. This is linked from our spreadsheet, but hopefully it's in my history here. And we can take a look. In fact, this is what our slides tell us to do. Go all the way to the end here. And we could take a look at this ChRNA7. So what I'm going to do, I'll come back to Excel in a moment, but ChRNA, go back to DNA subway, rather slip this and 7. When we search for it, thankfully it goes ahead and it filters down our results to just those genes. And if we look in the paper, there's a figure here where they talk about that. So here's a transcript, and one of the things that we see here is that in the mock sample, it's more highly expressed, and in the Zika sample, it is less highly expressed. So this is 709.1 transcript, and do we see that? It's this one. And if I just copy that, hopefully I copied it, and then go to our bootstrap, we should also be able to paste it in and hit enter, and also see that in the mock, it's higher, and the Zika, it's lower. Now, oops, I wanted to kind of go mock higher, Zika lower. There it says the estimated number of counts is between 400 and 500. If this is lawful, then maybe that's about the same thing, although we did not use all of the data that they used in this paper. We used a subset, but that's what you expect to see, that it's corresponding to the paper. If we look at another transcript where the reverse is true, the 7033.1, this one, fourfold difference. Look at the bootstrap. And we should see it's reversed, right? So that's excellent. But looking at genes one by one sometimes doesn't tell you the whole story. So although we could replicate that, the next thing we want to do is look at the ontology. So let me make sure I'm sharing my slides again. We replicated this to an extent, or at least just to get a feel. But the next thing is this thing called ontology. We're going to use this service, but first let me mention what that is. So an ontology is a way of grouping things according to their relationships, and also if those relationships are somewhat nested, then you can get some structure out of it. So if we just divided animals and plants, right, we could say that, well, maybe something is a tree or maybe something is a flower. And then if I told you that something is a lizard, then you would know that it's also a reptile, and that it's also an animal, and that it's also a living thing. So we develop, or some people, researchers have developed, a really nice way of taking everything that we know about every single gene and then putting it into a gene ontology. And sometimes you'll hear the phrase GO-term, G-O. And each one of these processes, like metabolic process, is extremely general. But then maybe it's something that's biosynthetic or organic molecule, and so on and so forth. And you can trace these ontologies and these relationships so that if you know that a particular gene has one of these activities, you could also see other activities it may be associated with. So the way that this works, to find out when you're looking at differential expression, one of your questions might be, well, what functions in the body are being, or in a cell, an organism, what functions are these genes that are really different? All of these genes here, right, are highly different in their expression values, and they're also significant. Do they have anything in common? Now, what I'm going to do is just take all of them, but you could take a subset. And oftentimes when you're a researcher, you may have an idea of certain genes you want to look at first, but for the fun of it, I'm going to take all of them, thankfully, because the service that I'm going to use is going to be quite fast, and see if we spot things where the word or the sort of thing we're looking for is to say is, are these genes overrepresented? Do they show up in certain processes more than you would think by chance? So I'm going to go ahead and open up my web browser back to this Go, which is linked, the Go Gene Ontology Shiny server, which is linked in the slides, or rather, yeah, in the worksheet as well. And it's really simple. I go to it and I can actually choose that this is working with human, and then I can paste in and then click submit. And it's going to take a few moments. But what it's going to try to do is figure out, given that list of genes, are there any biological processes where those genes are really involved? Now, many genes, there's some genes that will be activated no matter what you do, or involved no matter what you do. But what you're ultimately looking for, and you probably will end up narrowing down a list more than we have, are you looking to see, is there anything that makes sense? So in this case, it's telling you, giving you also the significance and talking about enrichment. Where do you see genes that tend to be in a certain pathway or tend to have a certain function? And a lot of these are coming out as being cell cycle and organelle and cellular localization. And in fact, if we go through and look at the different types of ontologies, there's one called the KEGG ontology. The reason why I like to look at that is because it narrows it down to some biological functions. And give it a moment to do that. So the top one here, 47 of our genes with the lowest, what we call false discovery rate, are all involved in cell cycle. And if you scroll down, here is a map of genes that are involved in the cell cycle. And all the ones that are in red are things that we're getting some hits for. So if you think about it, if all of these genes, not to say that they all are being affected in the same way, but if a lot of this graph was lit up versus some other graphs that could have been lit up, other different types of biological processes, it's a clue that maybe one of the things that's happening is affecting a cell cycle. Now, in the case of something like a viral infection, the trouble is that it's going to affect lots and lots of things in the cell. What you would hope, and then your work is kind of done, is that, for a particular thing you're looking for, RNA-Seq maybe has a very, very narrow effect, just a few genes that it affects very significantly. But here is pointing you direction. And in fact, if we go back to our slides, what we find is that in the paper that the data came from, they also implement cell cycle differences and dysregulation. And it's not surprising, because I believe that in this case, really you end up killing those cells and you're not able to produce the stem cells, and that's why you have microcephaly as one of the outcomes. So hopefully that was interesting for you. Looking back at the learning goals, to recap, I hope you got the hard details and the broad concepts down. Maybe those two things are counter to each other, but let's just stick to the high-level takeaway that an RNA-Seq experiment allows us to really interpret something about how gene expression may be happening in the cell. And we use high-throughput sequencing. In our case, we talked about Illumina to assess. To get those reads, we need to do oftentimes some cleaning to make sure we have good quality data. And then there's a toolkit, in our case, Callisto, which is our way of generating counts of reads and mapping them and normalizing them. And then once you have those counts, you can subtract away the variance that doesn't matter and use some statistics, which I didn't go into, but they're in the sleuth paper, so pay attention, you need them both, in order to explore those RNA-Seq results. And then you can use other methods like ontology enrichment to do more. So with that, we encourage you to go ahead and take a look at our website for other lessons that would be coming up and letting us know what you'd like to see. And it really has been a pleasure teaching RNA-Seq in this limited way, but hopefully got something out of it. And like I said, you can leave us some comments if you have additional questions. Okay, thanks.